Yeah, thank you for your introduction. I like to welcome all the listeners who joined this webinar, and we are glad to hold this webinar together with SERVA, our partner in the dialysis section. In the laboratory routine, daily routine, there are many applications where compounds have to be separated from each other. For example, purification of macromolecules if they have to be separated from undesired compounds. In protein purification, where you want to isolate your protein of interest and separate it from other cell compounds or other proteins in the sample. You may want to get rid of dyes or detergents. You want to change the buffer system to perform, for example, a pH shift. You want to perform binding studies in pharmaceutical or drug research area, or you may want to concentrate your sample. And recently, the cultivation of microbes or cell cultures in dialysis devices are possible. And this opens up a new kind of application. There are three methods for the separation by molecule size. The first one is dialysis, which is the topic of this webinar, and there are ultrafiltration and Gelfiltration too. And I want to talk about the Gelfiltration first, and want to say something about this method. It's a non-absorptive chromatography with an isocratic elution. It's distinguished between molecule sizes greater than 2 times, and therefore it's ideal for the separation of small salts or ions and larger proteins. The larger proteins elevate faster than smaller molecules, and as I said, it's ideal for desalting or rebuffering. It's a very simple process, and you can gain high recovery rates, and it's widely used. But there are some limitations of this method. The first one is that this process is related with sample dilution, and after the Gelfiltration you have to concentrate your sample again to go on with further analysis. And you need the ratio of the sample volume and column size is very small. That means you need large columns to separate higher sample volumes, and therefore you need an expensive equipment like a FPLC system. And the high throughput screening analysis are limited with this method. The second one, or the second method I want to talk about, is the molecule separation, is the ultrafiltration. Sorry, this separation takes place through a semi-permeable membrane under the usage of pressure, for example, in a centrifuge. It's a common used method for desalting and rebuffering, and for concentration of samples too. You need or standard laboratory equipment is You don't need a special equipment in the lab, and there are many different cutoffs available, and the handling is very easy. But there are some limitations too. That means due to the reduction of the sample volume, the membrane falling could occur, a sample could precipitate, and this results in losses of your sample. The third one I want to talk about is the dialysis. Dialysis is the diffusion of dissolved molecules across a semi-permeable membrane. The selectivity is determined by the pore size of the membrane, and the diffusion and dialysis occurs from a higher concentration to the lower concentration of the sample and the buffer. So in the image you can see a sample, a protein sample with an ionic contamination in a dialysis tube, which is in the dialysis buffer with a low concentration of the ions, and during the dialysis the ions pass the membrane and will be diluted in the sample. This process continues until the equilibrium. Dialysis is a gentle method for separation of proteins or other samples because there's no sample concentration on the membrane and low binding membranes are available. So therefore only a minimal fouling or cloaking occurs and the sample losses are minimal. There are a wide range of membrane types available and a wide range of cutoffs. And with modern dialysis devices, high throughput screening is possible. There are some parameters who influence the speed of the dialysis. For example, the temperature. You can see in this image below that the remaining amount of a dye in the sample will decrease with increasing temperature of the system. So that means that at higher temperatures the dialysis runs faster than at lower temperatures. But you have to check if your sample is stable at higher temperatures. So the larger the pore sizes of the membrane and the porosity is, the higher or the faster the dialysis speed is. A thicker membrane will decrease the dialysis speed. If you want to accelerate the dialysis, you should have a high surface area to volume ratio and short diffusion rates. Bigger molecules diffuse slower than smaller ones, and one important parameter is the concentration gradient between sample and buffer. And by changing the buffer you can keep a high concentration gradient, which is necessary for a high dialysis speed and a complete removal of the contaminants too. Among the well-known dialysis tubes, ready-to-use systems are available on the market, and they can be used out of the box, and no further preparation is necessary, and therefore you can save handling time. One of these ready-to-use systems are the Scienova Xpress Dialyzer, which I want to introduce on the next slide. Here you can see 4 types of our microdialyzers, and they are characterized by the sample volume. You can see the MD100, which for a volume of 100 microliters, the ED300 and MD300 for volumes up to 300 microliters, and the MD1000 for a sample volume of up to 1 milliliter. Due to unique geometry, fast dialysis can be achieved. It's ideal for small sample volumes. They are scalable from 1 to 96 samples. It's compatible with the SBS standard for microplates, and you can use it in liquid handling devices. On this image you can see a deep well plate where you can see cartridges of the microdialyzers, and up to 12 cartridges are put into one deep well plate. One cartridge contains of 8 segments, and one segment is for one sample each, and therefore you can use up to 96 samples in one plate. There are different cutoffs available between 3.5, 6 to 8 and 12 to 14, and in the next time we can deliver 220 and 140 kilodalton cutoffs. Now I want to show a video where the usage of a microdialyzer is demonstrated. Here you can see a cartridge of a microdialyzer MD100, and you can see from above 8 segments here, and you can load your sample with a standard laboratory pipet and can use the wide opening to fill in the sample. It's very easy, and you can use a multi-channel pipette too, as you can see here. And you can remove the sample on the same way, the opposite, and it's very easy. The dialysis buffer is filled in each well of the deep well plate, and after that you can put in the microdialyzer and start the dialysis. Next I want to show you some typical applications for the dialysis. The first one is desalting, as you can see on the left side. There you can see the removal of PBS in the sample, and you can see that this process occurs very fast, and within 2 hours you can remove the salt in the sample completely. On the right side you can see the dialysis of a BSA sample with detergents, SDS and caps. You can see that both compounds were dialyzed and removed from the sample, but it takes a much longer time than the desalting process, which you can see on the left side. To save handling time, we put together a kit where you can use one reservoir of the buffer instead of 96 wells of a deep well plate. If you use same samples and the same buffer and you want to dialyze it, you can use this system, and you can save handling time. And I want to show you a short video where you can see how it works. Yeah, you can use it on a magnetic stirrer, and you can mix the buffer during the dialysis. You can load the sample like you are using a deep well plate. Then you put the grid with the dialyzer into the buffer, dialyze it, and after that you can remove a complete grid with the microdialyzer and go on with removing the sample and your further analysis. As you can see in this diagram, the dialyzer speed is faster than in samples who are not without mixing the dialysis buffer in deep well plate. You can see this black line is the dialysis in a deep well plate. The red line is the dialysis in this dialytic magnetic mixing box. One typical application is the reactivation of a protein or enzyme after it was inactivated by using, for example, urea in a purification step. So in this example we show the activity of trypsin, and on the left side you can see the concentration of urea, and you see how the urea is removed from the sample during dialysis. It's a very fast process, and you have nearly complete removal of urea within 2 hours. And on the right side you can see the activity of trypsin, and here you can see the activity in the presence of urea, and the third one you can see, of third and fourth one, the activity without urea, and in the second one you can see the activity after the removal of urea, and can see that yeah, a lot of the activity is regained after the dialysis. Now I want to introduce a new kind of dialyzer, which we which is characterized by that you don't have to remove the dialyzer from the deep well plate if you want to fill or remove the dialysis buffer in the outside. As you can see here, there are two openings of this in this dialyzer, and the first one is for loading and removing the sample in the inside of the dialyzer, and the right one is for removing or loading the buffer in the outside. So there's a little channel in the dialyzer where you can which you can use for the buffer. It's you can use up to 300 microliter sample, can use up to 96 samples in each plate, and it's automation compatible too, which I want to demonstrate in a short video. You can see this ED300 in an 8-volt liquid handling device. You can see the deep well plate with the ED300 cartridges, and you can see how the device is loading the buffer now to the one opening, and after that the sample is loaded to the cartridge, and this now you can see the removal of the sample and buffer again. It's very easy, and you can see now the buffer from the outside with a light yellow color, which means that the dye was dialyzed in the cartridge. Okay. So this cartridge is for the usage of a equilibrium dialysis, for example, of the determination of free cortisol, which is important in medicine, where you have to determine the amount of cortisol in the serum and the amount of free cortisol in the serum. And as you can see in this illustration, in the serum there are both the bound cortisol and the free cortisol, and during the equilibrium dialysis the free form is passing the membrane, and in the equilibrium you have the same concentration of the free cortisol in the inside and outside of the dialyzer. And on the next slide you can see the curve of the dialyzer, the cortisol concentration, and you can see that the equilibrium is achieved, and Now, determination of the cortisol concentration, of the free cortisol concentration is possible. And in combination with the usage of a liquid handling device, can use it with high throughput screening or in standard laboratory applications.